Ladies and gentlemen, it's a great pleasure and a privilege to welcome all of you to the 2021 Nobel Lectures in Physiology or Medicine. My name is Ole Petter Ottersen. I'm the president of Karolinska Institutet. During the Nobel Week, we celebrate science and we rejoice at the unique contributions of some exceptional individuals whose discoveries, in the words of Alfred Nobel, have conferred the greatest benefit to humankind. Within physiology or medicine, the ultimate goal is to achieve scientific breakthroughs that significantly deepen our understanding of health and disease. The discoveries we celebrate this year are stellar examples of such breakthroughs. Today, it's an honor for me to extend my warmest congratulations on behalf of Karolinska Institutet to David, Julius, and Ardem Patapoutian. They share the 2021 Nobel Prize in Physiology or Medicine for the discoveries of receptors for temperature and touch. This morning here in Stockholm, the temperature had dropped to minus 17 degrees Celsius. When I was walking to work, I felt very clearly that we humans, we do have receptors that react to temperature. So it will be very exciting and also, I would say, very timely to hear more about the important findings of our Nobel laureates today. Without further ado, I now give the floor to Abdel El-Manira, Professor of Neuroscience and a member of the Nobel Assembly, who will introduce this year's laureates. Please, Abdel.
President of the Karolinska Institutet, esteemed laureates, dear colleagues and friends, ladies and gentlemen. On behalf of the Nobel Assembly at the Karolinska Institute, I welcome you all to the 2021 Nobel Lectures. I have the great pleasure and honor to introduce this year's Nobel laureates in physiology or medicine, Dr. David Julius and Dr. Ardem Patapoutian. They have been awarded this year's Nobel Prize in Physiology or Medicine for their discoveries of receptors for temperature and touch. The philosopher Immanuel Kant wrote, "All our knowledge begins with the senses, proceeds then to the understanding, and ends with reason." Our senses are not only the source of reason, but they are also the essence of our survival. They underpin our interaction with the world around us. This year's Nobel Prize pertains to our ability to sense temperature and touch. These sensations are omnipresent. We cannot switch them off at will. We take them for granted in our daily lives. And yet, the absence of these sensations has detrimental consequences for our well-being. The question then is, how temperature and touch are sensed and converted into electrical signals in the nervous system. This question remained a mystery until the work of this year's Nobel laureates. They have discovered novel classes of molecular sensors that respond to heat and mechanical pressure. These sensors convert physical stimuli into electrical impulses that are then conveyed to the brain to allow us to perceive temperature and touch. Nature not only provided the mystery, but it also afforded us the tools to solve it. David Julius took advantage of capsaicin, a natural chemical compound found in chili peppers that induces a burning sensation, to identify a novel receptor protein named TRPV1. Remarkably, David Julius found that TRPV1 is not only a receptor for capsaicin, but it is also a sensor for heat. This was a major breakthrough, leading the way to the unraveling of additional temperature sensing receptors belonging to the TRP channel family. Each of these receptors is activated over a specific temperature range, and together they act as a biological thermometer that allows us to control our core body temperature and to distinguish pleasant warmth or a cool breeze from painful heat. Ardem Patapoutian took advantage of a cell line with inherent sensitivity to mechanical stimulation, and using a painstaking gene silencing experimental analysis, he succeeded in identifying the gene encoding the elusive mechanosensitive channel that he named piezo, after the Greek word pressure. Piezo channels provide us with the sense of touch, to feel the texture of objects, and to monitor the position of our body in time and space, our sixth sense called proprioception. The work of this year's Nobel laureates has unlocked one of the secrets of nature. Their discoveries of receptors for temperature and touch have fundamentally changed our views on the functioning of sensory systems and enhanced our understanding of the complex interplay between the environment and our senses. At this point, reason obliges that I give way to the Nobel laureates, who will now give us a true sense of their seminal discoveries. The first lecture is on the discovery of receptors for temperature by Dr. David Julius. David Julius is a professor and chair of the Department of Physiology at the University of California, San Francisco. He holds the Morris Herrstein Chair in Molecular Biology and Medicine. Professor Julius was born in Brooklyn, New York, where he attended public elementary and high schools. He receives his bachelor's degree in life sciences from the Massachusetts Institute of Technology in 1977, and obtained his doctoral degree in biochemistry in 1984 from the University of Berkeley, California, Berkeley, where he worked with Jeremy Thorner and Randy Sheckman on peptide hormone processing and secretion in yeast. Professor Julius performed his postdoctoral training in the laboratory of Richard Axel at Columbia University, New York. During this time, he developed novel expression cloning methods, enabling him to identify genes encoding serotonin receptors. And he joined the faculty at the University of California, San Francisco in 1989. David Julius is a member of the National Academy of Sciences and the National Academy of Medicine and the American Academy of Arts and Science. David Julius will now deliver his Nobel lecture entitled From Peppers to Peppermints, Insights into Thermosensation and Pain.
Hello from San Francisco. I'd like to begin by thanking the Karolinska Institute and Professor Otteson for hosting these lectures. And I'd like to thank the Nobel Foundation for their creativity in putting this hybrid celebration of Nobel Week together, which at least allows us to enjoy one another's company in some form during this still challenging pandemic times. And of course, I'd like to thank the Nobel Assembly for bestowing this tremendous honor upon me and my co-laureate, Ardem, with whom I'm very pleased to share this prize. And in a larger sense, I'd like to thank the Nobel Assembly for recognizing the field of pain and somatosensory research, which has brought great excitement to the field, to our laboratories, to our institutions, and even to the many patients around the world who suffer from chronic and persistent pain syndromes. So that's actually a good place to start, because much of what our motivation for much of what we've learned over the last few years has been to ask questions about pain and pain mechanisms and understanding pain. So pain, of course, is a submodality of somatosensation, the sensation that we colloquially refer to as touch. And I often argue that among our five senses, somatosensation and pain is probably the most important for our survival and well-being. And that's because pain serves as a main warning and protective system that tells us when we have or are about to experience bodily injury and to initiate appropriate protective reflexes. And in people in whom this system is rendered inoperable due to genetic mutations or diseases, those individuals are at great risk of bodily injury or death. The problem is, of course, is that while pain is incredibly useful and necessary as an acute warning system, it often outlives this phase and instead becomes persistent and debilitating. And the goal is to understand this switch between acute and chronic pain, ultimately with the hope of preventing or reversing that. Now, another challenge, I think, in the pain field in the long run is to better define different pain syndromes, to ask how different chronic pain syndromes differ cellularly, molecularly, in terms of their circuitry, and to understand what the underlying molecular mechanisms are, because I think until we do that, we won't be able to adequately treat different pain syndromes. So let me begin by just giving a very brief overview of the pain pathway. Of course, pain, like other sensory systems, involves the detection of peripheral stimuli, environmental stimuli, as well as some endogenous stimuli. And this occurs at the tips of primary afferent or sensory nerve endings that transduce this information then to the central nervous system, first to the spinal cord, and ultimately to the brain, where we perceive what's happened peripherally as a noxious or painful stimulus. In my lab, we focused mostly at understanding what happens here where pain begins at the periphery in these primary afferent nerve terminals, and which in aggregate are really remarkable cells. And why are they so remarkable? Because they're tasked with the job of being able to detect a wide range of stimuli that include physical stimuli like changes in pressure and temperature, which is really sort of the focus of the lectures you'll hear today, as well as other things in our environment, such as noxious chemical agents, as well as agents that are produced in our body as a consequence of tissue or nerve damage. And what's remarkable about these sensory nerve endings is that they have mechanisms, receptors, for sensing each and every component of the so-called inflammatory soup. And of course, the purpose of this is to heighten the sensitivity of the primary afferent nerve terminal as part of the guarding response to tell us when we have experienced injury so that we can protect that area. So it really changes the gain of the nerve fiber. And the goal in my lab, and many labs in this area, is to understand how all these different stimuli focus in on the primary afferent nerve terminal, including physical stimuli and chemical stimuli, to regulate its excitability under normal conditions and under conditions following injury. Now, studying these mechanisms, especially 20 or 25 years ago when we began this, was somewhat challenging because the genetic and other tools to identify molecules and mechanisms involved were lacking. And so we turned to an approach, and that is to really exploit natural products and in some sense folk medicine to use tools, pharmacological tools from these systems to try and identify and probe mechanisms that are involved in pain sensation. And I sort of refer to this as pharmacology honed by evolution. And I've always been fascinated by this because studying natural products in folk medicine is really sort of the nexus where human behavior, chemistry, and neurophysiology come together. And I think some of the examples that have really inspired me and other people in this field come from some of the following individuals. For example, Saul Snyder's brilliant use of morphine and other opiate alkaloids to discover opiate receptors. The work of Sir John Vane, who explored the role of aspirin and salicylic acid as a product of the willow bark, which we know is an analgesic, and through that route discovered the existence of cyclooxygenases and prostaglandins. And then Raphael Mechoulam in Israel, who asked what the psychoactive ingredient in marijuana is, and identified THC as the active psychotropic component, and then went on to identify endogenous endocannabinoid-like compounds, namely anandamide and 2-AG, through this kind of route. I've always been fascinated by this approach, and I think this has served as inspiration for many of us who have taken this route of exploiting natural products in folk medicine as a way to explore endogenous mechanisms of neural signaling or even cardiovascular signaling in human physiology. And I would add that what's really beautiful about this approach is that if you think about it, these agents still represent the foundation of the two main groups of analgesic agents that we use today, namely opiate analgesics and nonsteroidal anti-inflammatories. So this is a very powerful approach. Now, we've sort of turned this coin over on its other side, and instead of asking how natural products work to deliver analgesic relief, we've asked how natural products work that serve to induce pain, that serve as irritants. And the granddaddy in this field is really capsaicin, the main pungent ingredient in chili peppers. But there are also other plant-derived irritants, including menthol from mint leaves, and these compounds, the so-called isothiocyanates and thiosulfonates, that represent the pungent irritants in wasabi, members of the mustard family of allium plants such as onions, shallots, garlic, etc. Now, in this regard, there are really two groups, when I went back and thought about this, whose work really influenced our approach, specifically in regard to using these plant-derived irritants. And one of these is the brilliant work of Jansko and colleagues, together with his wife, Aurelia Jansko-Gabor, whose team were really the first to use capsaicin at the University of Szeged in Hungary as a chemical probe for a subset of sensory neurons that turned out to be very important in pain sensation. So they showed in the 1950s and 60s that exposure, that capsaicin served as a very selective excitatory agent for a subset of primary afferent nerve fibers that are dedicated to pain sensation, the so-called nociceptors. And they further showed that exposure of sensory neurons to capsaicin, especially at high doses, led to a functional desensitization of these cells, as well as ultimately to their death. And furthermore, they showed that animals injected with a bolus of capsaicin would show a very profound decrease in their core body temperature, which is probably why we like to eat hot peppers in hot climates, because it leads to a centrally mediated, hypothalamically mediated cooling of the body through vasodilation and sweating. But that also suggested that whatever the mechanism of capsaicin might be on the primary afferent, that this somehow was involved in sensing external temperature and reporting this information to the CNS. The other group that greatly influenced our work was that of Yngve Zoderman, together with Herbert Hensel, who did that work actually at the Karolinska Institute. Here's a picture of Zoderman in 1926, speaking with Lord Adrian outside the Karolinska Institute during a physiological conference. And what Hensel and Zoderman showed was that menthol has a very specific action on a subset of nerve fibers in the cat tongue, and that the actions of menthol as an excitatory agent could be suppressed by warming or enhanced by cooling. And together, these observations suggested that the psychophysical effects of these natural products are mediated through the selective action on somatosensory nerve fibers. It also suggested later on to many of us that if one could define the molecular targets for these natural products, that this would reveal some important endogenous mechanisms of pain and/or temperature sensation. So later on, the focus really in ensuing years was really on understanding how capsaicin works. In about the 1990s, a number of scientists, particularly at the Sandoz Institute at the University of College London, began to ask how capsaicin works from a biophysical perspective. So they patch clamped sensory neurons, and they had data to suggest that what capsaicin does is to enhance the permeability of the membrane of sensory neurons to both mono and divalent cations. And the presumption was that capsaicin acts upon a receptor and/or an ion channel that mediates these effects, and that the selective actions of capsaicin might be a consequence of the expression of such a receptor on these cells. Although I have to say at this time, there are also competing ideas that capsaicin works more non-selectively by integrating into the membrane and somehow forming some kind of an ion permeation pore. But this set up a number of questions, obviously, in the 80s and 90s that harken back to the questions that were raised by Jansko and by Hensel and Zoderman, and that is, do natural irritants target specific sites on sensory nerve fibers? What's the molecular nature of these putative receptors, if they do exist? And if they do exist, can we use these as molecular tools ultimately to ask if they mark functionally distinct sensory neuron subtypes? In particular, are they good probes for marking subsets of neurons, the presumptive nociceptors that are involved in either temperature or pain sensation? And really the biggest question is, what are the endogenous physiological roles of such molecules if they exist? Obviously, we don't have these putative receptors in our body just so we can experience the culinary wonders of spices. What are their roles in normal physiologic circumstances? And this really set up what we often refer to as the holy grail of molecular pain research at the time, which was to find the capsaicin receptor. And this grail was really reached when Mike Caterina joined my lab. He's now at Johns Hopkins Medical Center, when he took on the challenge of cloning what we hoped would be a gene encoding the capsaicin receptor. And he had a very simple approach, which is to take somatosensory neurons from a mouse or a rat and generate a cDNA library, expression library from these cells en masse, and then to introduce these into non-excitable, non-neuronal cells, in this case, so-called HEK 293 fibroblasts. And the idea was that if capsaicin really acted on some kind of a receptor or a channel that increased intracellular calcium, as people like Humphrey Rang and Stuart Bevan had suggested, that we could detect this with the calcium-sensitive dyes developed by the late Roger Chen. And so what we did was load these cells with the calcium dye called Fira, introduce capsaicin, and we looked for cells that glowed, which presumably had taken up a cDNA clone that allowed them to express a receptor that rendered them sensitive to capsaicin. And indeed, Mike was able to do this, and he succeeded in this. This was somewhat, of course, of a eureka moment. And he showed through sequencing and together with Makoto Tominaga and others in my lab that this gene encoded, in fact, an ion channel that has permeability to both mono and divalent cations. A couple of years later, and I should also say that, that pharmacologically what Mike was able to show is that, in fact, this receptor is a great reporter for pungency. So if you ask how this receptor responds to extracts from chili peppers, ionic currents go through this receptor, respond in direct proportion to the perceived pungency of the chili pepper, using capsaicin as a standard. And so in essence, we now had a molecular reporter that was a substitute or a mimic of the so-called Scoville units, which are classically used to report pungency. Now, a couple of years later, Dave McKemmy joined my lab, and together with Werner Neuhauser, asked the same question for menthol. Is there a receptor for menthol, and can we use the same function-based cloning approach to identify that? And indeed, Dave and Werner were able to identify a cDNA encoding a menthol-sensitive receptor. Now, I think one of the most beautiful outcomes of this part of our work was really to find that there's a beautiful molecular convergence and logic here, and that is that both of these receptors turn out to be members of the same molecular family, the so-called family of TRP ion channels. The menthol receptor, TRPM8, and the capsaicin receptor, TRPV1, are molecular cousins by belonging to this family. And in fact, the receptor for the so-called wasabi receptor that's activated by these other agents, pungent agents, the isothiocyanates and thiosulfonates, also belong to this family. And so we see this beautiful example of convergence of mechanism for these natural product irritants acting on these channels, which really illustrates the importance of TRP channels in sensory processes at large, but in somatosensation and pain specifically. Now today I don't have time to talk about TRPA1, the wasabi receptor. This is a very interesting channel that plays a major role in chemonociception, that is our ability to detect environmental and endogenous chemical irritants, and hopefully if I come to Sweden next year, I can tell you more about work that we and others have done in this area. But for today, I'm going to focus on the capsaicin and menthol receptor. And the real question here, of course, is what is the natural role of these ion channels in physiology? And this question was really answered when Mike and Makoto and others in my lab found that the expressed channel, whether you express it in a fibroblast, in a frog oocyte, whatever, has intrinsic sensitivity or renders the cell sensitive to heat. And as you can see here in this beautiful temperature response curve, the salient characteristics of this response is that the channel shows this very steep sensitivity to an increase in temperature, shown here on the x-axis with ionic current on the y-axis, and it shows a temperature coefficient of activation greater than 20. For most ion channels, this would be no more than 1 to 3 in that range. So it has a very steep cooperative activation by heat. And the other thing is that there's a very defined threshold at about 43 degrees, which is actually in keeping with our psychophysical threshold for when we can discriminate between an innocuous warm stimulus and a noxious heat stimulus. Conversely, as Dave and Werner showed, the menthol receptor is activated by cold. And so that channel opens as the temperature drops below about 25 degrees, and also shows this high temperature coefficient of activation, not quite as high as TRPV1, but still higher than most standard ion channels that you would look at. And again, the threshold at about 25 degrees corresponds beautifully to where we would discriminate between something that's warm and something that's cold. So this is really the threshold for psychophysical discrimination of cooling. So this sets up the idea that these two channels serve as little molecular thermometers that allow our sensory nerve fibers to report changes in ambient temperature in the hot and cold direction. And when you come in contact with the natural product agonists or allosteric modulators of this channel, namely capsaicin or menthol, what they do is they lower the activation threshold for the channel and allow the heat-activated channel to open at a lower temperature and the cold-activated temperature to open at a warmer temperature, in essence serving as a chemical psychophysical mimic of a hot or cold experience, respectively. And this model lays out at least two predictions. One is that these ion channels are intrinsically temperature sensitive, that they themselves, that their biophysical properties themselves describe their sensitivity to temperature, and perhaps most importantly, that they actually contribute to an animal's ability to discriminate ambient temperatures in vivo. And the first prediction was beautifully borne out by the work of Erhu Kao in my lab, now at the University of Utah, when he purified the channel to homogeneity, reconstituted this into a totally synthetic lipid bilayer, and then used patch clamp analysis to ask what happens when you expose this patch to a quick burst of radiant heat with an infrared laser. And what he showed, together with our collaborator Feng Chin at the University of Buffalo, is that this leads to beautiful activation of the channel, as you can see in these characteristic outwardly rectifying ionic currents in a temperature-dependent manner. And in fact, if you look at the biophysical and thermodynamic properties of this response, in terms of the temperature threshold, the activation rate, the temperature coefficient of activation, it beautifully matches the parameters that we and others, including Feng and Peter McNaughton, have measured in transfected cells expressing this receptor, and indeed even in sensory neurons that are heat sensitive. So I think this fulfills one of the beautiful predictions that, in fact, these proteins themselves are little biophysical machines that act as molecular thermometers. Now, what about in vivo? I think the most elegant demonstration of this comes from an experiment that Diana, Jan, and Sven carried out when they were in my lab, where they made a knockout mouse in which the menthol receptor has been deleted from the genome. And here's a wild-type mouse in this very simple paradigm where we ask if it can discriminate between a warm platform at 30 degrees and a colder platform at 20. And as you can see, even without any training, this mouse prefers, as we would, to sit on the nice, warm, comfortable side and will not even venture onto the cold side. But if you look at an animal in which the menthol receptor has been deleted from the genome, a TRPM8 knockout mouse, you can see that that animal really loses its discriminatory ability and can no longer discriminate a warm from a cold surface and will wander freely over this surface and spend equal amount of time in both ends. And you can continually lower the temperature of the cold plate until you get to very noxious temperatures of about 5 or 0 degrees, and only at that point can the animal then discriminate between the warm and the cold surface. And this little chamber is actually our donation to the Nobel Museum, which we just sent off this morning. So I think that experiment really beautifully shows that, that these receptors, specifically in the case of tryptamate, are required for an animal's ability in vivo to discriminate between ambient temperatures. Now, with molecular probes in hand, this is often, as is true for all of the sensory systems that have been looked at, olfaction, taste, etc., this now allows us to begin to ask at least some rudimentary questions about coding. And what Jan Siemens, when he was in my lab, could show with antibodies against TRPV1 and tryptam8 is that while there's some overlap, for the most part, these molecular markers decorate different non-overlapping cell types. They're all small diameter, so they're of the sort of nociceptive C and A delta category, but they do discriminate among cells within that, marking subpopulations. And so what this suggests, at least to the initial degree, is that in fact there is some sense of labeled line logic in the somatosensory system, at least with respect to hot and cold discrimination. And this really holds up the predictions of Sherrington and then later Ed Pearl, who suggested that cellular and behavioral specificity begins at the primary afferent and not just at the spinal cord, that there is specification initially where signals are detected. Now, of course, that doesn't say that there isn't. There must be integration to the level of the spinal cord, and in fact, work from Mark Hoon and Gary Lewin have suggested that our ability to sense warmth and understand what that means is really an interaction between the hot and the cold fibers and the integration of this information at the level of the CNS. Now, while we've identified TRPM8 and TRPV1 as being important players in this regard, over the last few years there are a number of other TRP channels that investigators such as Peter McNaughton, Thomas Votes, and others have implicated as temperature-sensitive TRP channels, and where and how along this spectrum they contribute to our ability to sense hot, warmth, or cold is still an area of intense investigation. But I think, needless to say, TRPM8 and TRPV1 are not the only players in this area, and they collaborate likely with these other channels to set boundaries for discrimination of hot, cold, and warmth. And as I said, the interplay between TRPV1 expressing fibers and TRPM8 expressing fibers likely also helps set our sensitivity for temperatures at the midline. Now, before leaving the area of, or this topic of talking about mechanisms of acute temperature sensation, I just wanted to mention some work that we've done that really speaks to an area that we're also interested in, which is the evolutionary adaptation of sensory physiology. And as we all know, sensory systems are prone to rapid evolution to suit lifestyle and niche behaviors of different organisms on the planet. And in a really beautiful series of studies, Elena Gracheva and Julio Cordero Morales from my lab looked at a really interesting aspect of this in animals that carry out what we colloquially refer to as infrared sensation. So that includes certain species of snakes, like pit vipers, and there's one mammal that is credited with having this sort of sixth sense of infrared sensation, and that's the vampire bat, whose sanguinary lifestyle necessitates that it be very effective at finding the blood supply in its victim, say a sleeping cow or a pig. And to do this, it uses a part of its nose that I'll show you in a minute that's highly fenestrated with trigeminal sensory nerve fibers that are acutely sensitive to heat. And Elena and Julio ask whether there are molecular adaptations in this animal that facilitate its ability to carry out this process. And what they showed is that actually the vampire bat expresses two distinct splice forms of the TRPV1 ion channel, what we call the short and a long splice form. And what they showed is that the short splice form is specifically expressed in trigeminal ganglia, whose nerve fibers innervate these little leaf areas, the so-called pit areas on the snout of the vampire bat, which is what it uses to detect blood flow and heat source in its victims. And then there's a long form, which is more like the form that we possess, that is expressed in the dorsal root ganglia of these animals and innervates everything from the neck down, namely its trunk, which presumably subserves the same purposes that it does in our body to detect noxious heat and help regulate core body temperature. And now another beautiful example in the TRPM8 side was shown by Ben Myers when he was in my lab, now at the University of Utah. And what Ben asked was whether TRPM8 channels show differences in their thermal activation thresholds in animals whose core body temperatures differ. And for this, he looked at birds, which have a higher core body temperature than mammals, and then in cold-blooded animals and frogs, whose body temperature is on average lower, depending upon their ecological niche. And what Ben showed beautifully is that, in fact, the temperature response curve for TRPM8 is, in fact, in keeping with the average core body temperature of these animals, again showing this beautiful example of evolutionary adaptation that shifts the thermal threshold of all of these channels in keeping with an animal's niche and lifestyle needs. Now, understanding acute pain sensation and acute thermoreception is clearly a very important and fascinating subject, but from a clinical perspective, it's also really important to understand how the sensory nervous system and how the somatosensory system adapts to injury. Because we all know that pain thresholds change after injury. If you get a sunburn, for example, or some other inflammatory episode, sprain an ankle, or have osteoarthritis or rheumatoid arthritis, our sensitivity to pressure and temperature and even chemical irritants can become greatly heightened. And this hypersensitivity underlies, both peripherally and ultimately centrally, the genesis and the development of many persistent chronic pain syndromes. So a very important goal in our lab and in many labs in this field is to understand the changes in the chemical and in the physiological and chemical environment that drive these changes that lead to pain hypersensitivity and to changes in gain of the primary afferent that engender these sort of long-term changes in sensitivity of the pain pathway. Now, in this regard, TRPV1 has become a very important target for this and focus of study into understanding how tissue injury leads to an increase in sensitivity of the primary afferent nerve terminal and ultimately behavioral sensitivity. And that's because we and many other people in the field have come to understand that TRPV1 is modulated by a variety of the components of the inflammatory soup that I discussed in the initial slide. And what we've come to understand about TRPV1, and in fact many other members of the TRP channel family, is that these are very sophisticated signaling machines that serve as polymodal signal integrators. And with respect to TRPV1 and pain, there are two sort of relevant categories of modulators. There are direct allosteric modulators of the channel, namely extracellular protons and endogenous lipids such as anandamide and N-arachidonyl dopamine that get produced during tissue injury. And Makoto Tamanaga and Sven Hjort in my lab have investigated the mechanisms which by extracellular protons affect the TRPV1 channel. And what they showed, in keeping with early studies from Piero Geppetti and Peter Ree and others looking at the effects of protons on sensory neurons, is that what these agents do and what protons and these other lipids do is really to shift the thermal threshold for activation for TRPV1, enabling the channel to open at cooler temperatures. And you can see this here when the channel is expressed in either oocytes or mammalian cells. Under normal pH conditions, the channel opens with this very concerted temperature response threshold of about 40 or 43 degrees. When you acidify the extracellular surface of the cell, you see two things happen. The threshold for activation shifts way back closer to body temperature, and at suprathreshold temperatures, one often sees a response of greater intensity. The other way in which this channel is modulated through components of the inflammatory soup that interact not directly with the ion channel, but on their own receptors. This would include molecules like bradykinin, extracellular nucleotides like ATP, or even neurotrophins that act on their own receptors and through downstream second messenger signaling systems, such as phospholipase C and protein kinases, modulate the sensitivity of the channel and again shift its sensitivity to heat to lower temperatures. So these pathways, both the direct modulation of the channel by allosteric modulators like protons and lipids, and the indirect modulation by neuropeptides like bradykinin or nerve growth factor, are really important parts of the pathway by which the elaboration or components of the inflammatory soup modulate the sensitivity of the nerve terminal and change its gain, particularly for temperature. And so this raises the idea that these channels that there's some therapeutic value in targeting these channels in the periphery for treating a variety of persistent pain syndromes, particularly those associated with inflammation that generate these and other components of the inflammatory soup. And in fact, over the years, a number of antagonists for TRPV1 have been developed. And in this early study from Mike Robotham, what he showed is that volunteers who have taken one of these drugs called ABT-102 show, as you might expect, and in a really beautiful, I think, validation of the role of TRPV1 in humans, they show an increase in their in the thermal threshold at which they can detect a noxious heat stimulus. So what that means is that their ability to detect something that's noxiously hot requires that object or whatever they're touching be at a higher temperature. So what that means is that they're drinking a cup of coffee, it's harder for them to discriminate that as a noxious stimulus because their threshold for detecting noxious heat has now been increased by a significant amount, in this case about 4 or 5 degrees. So this beautifully validates the role of TRPV1 as an acute heat sensor in humans. But of course, it raises the problem in that what it does is it erodes the protective capacity of the pain system to tell you when you have or are about to experience damage. And this is something that really, of course, concerns drug development. And I would say not just for TRPV1 antagonists, but for any other new type of analgesic. An important goal, of course, in analgesic therapy is to prevent the generation of persistent or chronic pain under so-called maladaptive conditions without eroding the protective function of the pain pathway. And moreover, what's been shown in beautifully hearkening back to the early studies of Jansko and colleagues is that these TRPV1 antagonists also produce a subtle and transient hyperthermia in a number of individuals who take them, again, attesting to the fact that TRPV1 positive fibers are important for reporting ambient temperature to the central nervous system and eliciting changes that control core body temperature. But these kinds of on-target side effects have raised issues about the use of these drugs as analgesics. And I think what we really need is to consider the development of drugs that have a more nuanced effect on TRPV1 and other channels that interfere with the ability, for example, of inflammatory agents to modulate the channel, but that do not shut down their core function of detecting so-called adequate or acute signals that we need to protect ourselves. And in this regard, a really important goal from our lab and others is to understand what these biophysical machines look like in greater atomic and functional detail. Because then I think we have a greater chance of developing drugs through some rational mechanisms that can, for example, interfere with the ability of specific and key inflammatory agents for modulating the threshold of activation of the channel without completely turning off its ability to detect noxious heat. And so the goal here is to really take this cartoon and turn it into a 3D atomic structure that tells us where all the nooks and crannies are in this channel that are involved in gating transitions to help us think more about designing drugs. And when I mean the community of pharmacologists that have more subtle effects on these channels. And this goal was really achieved a number of years ago by a fantastic interaction that we've had with my friend and colleague at UCSF, Yifan Chang, and members of his lab, and in particular, Erhu from my lab, who joined up together with Maofu Liao from Yifan's lab a number of years ago with the challenge of identifying or characterizing an atomic structure for any TRP channel, but in particular TRPV1 because of its iconic involvement in pain sensation. And Erhu's now, as I said, at the University of Utah, and Maofu has his own lab at Harvard. So a number of years ago, they were able to achieve this goal by using electron cryomicroscopy to solve the three-dimensional structure of TRPV1, and since then we've used this to solve the structure of numerous other TRP channels in the pain pathway. And this really was another eureka moment because we were part of hearkening in what now is referred to as the resolution revolution in cryo-EM, whereby, particularly for membrane proteins, could be taken to atomic or near atomic resolution using this technique. And one of the beautiful things, as you may know about cryo-EM, is that when you deposit proteins on a grid, so here are a bunch of individual TRPV1 ion channels on a grid. I really think this is just an unbelievable picture because you can actually look at the molecule with its central ion permeation pathway. Each one of these looks like a little donut sitting on a grid, and it's looking at the molecule. You know, seeing is believing, and it's looking at the molecule in real life. But one of the great things about cryo-EM is that when these molecules are deposited on a grid, they can be found in many different conformational states. And in particular, hopefully from closed to open. And so this affords one the possibility of seeing a protein when it's actually going through the life cycle of its conformational transitions. And if you have ligands that can bias this equilibrium, then you can really hope to define different conformational transitions, as in this case, a channel goes from closed to open. And of course, the TRPV1, I think, represented an incredible opportunity to do this, because among TRP channels, it's the one that really enjoys the richest pharmacology. We have capsaicin. We also have this compound originally characterized by Peter Blumberg at the NCI, resinifertoxin, which again is another natural product from a flowering cactus in the Mexican highlands. That's an extremely potent vanilloid agonist. And then there are other things on the planet that defend themselves by activating pain-sensing nerve fibers, and this includes animals like scorpions and spiders. And when Chris Bolin was a student in my lab, he identified this 80 amino acid peptide, which we call double knot toxin, from a spider called the earth tiger tarantula. Its name is very descriptive of what it's like if you get bitten by it. And we've been able to use all of these probes, and double knot toxin binds to TRPV1 and stabilizes it in its open state with very high avidity. So taking advantage of these tools, we were able to see TRPV1 in all its glory in its three-dimensional atomic structure. And here's a ribbon diagram of that structure that shows that the channel is a homotetramer. And without going into details, I just want to say that it's a distinct, it's a distant molecular cousin of voltage-gated potassium channels, and has the same sort of quaternary structure that Rod McKinnon famously described years ago for bacterial and voltage-gated potassium channels. And we were able to see this channel not only in its closed form. I should say one of the exciting observations was that whereas voltage-gated potassium channels have a restriction in the ion permeation pathway down here, the so-called cytoplasmic gate, we saw that TRPV1 has two restrictions in it, one up on top near the selectivity filter of the ion channel, where the ions go through the central pathway, and one down here closer to the cytoplasmic face that's akin to the gate, the main regulatory gate seen in voltage-gated channels. And we could determine whether these gates are actually operable gates that control movement of ions by looking at the channel in its fully open form, which we were able to do by giving it resinifertoxin, shown here, and this spider toxin to lock the channel into its fully open conformation. And indeed, when you do that, you can see that these restrictions are now relieved in the channel, showing that they are operable control points. And the idea here is that these dual restrictions are what underlie the ability of this channel to serve as a polymodal signal integrator and to have this very rich physiologic modulation by various components of the inflammatory soup. And what this really does is give us an opportunity to understand how these channels work and to look at sites that might be better sites for regulation of the channel to develop analgesic drugs. And in one brief example that I'll talk about, I just want to show how understanding the structure of these channels does provide some insight into how one important pro-analgesic agent, namely extracellular protons, works to regulate the channel. So Sven and Makoto, when they were in my lab, showed that there is an extracellular glutamate residue, this E600, that serves as a main locus for titration by extracellular protons and is required to enhance sensitivity of the channel to heat under acidic conditions. And if you look at the structure in its closed versus its open state in this extracellular loop, what you can see is this glutamate residue is held in close apposition to neighboring loops in the channel through ionic interactions. And in the open state, these interactions are pulled apart, suggesting that what protons do is to salt these interactions out and pre-break these bonds to make it easier for the channel to open, and that this may be the mechanism whereby protons facilitate opening of the channel and lead to this shift in thermal threshold and this and somewhat loss of cooperativity in activating the channel by heat. And indeed, in recent structures resolved by Kaiwa Zhang in collaboration with myself and Yifan, he could look at TRPV1 channel under different pH conditions, namely lowering the pH from pH 7.4 to pH 6, and even as low as 5, and could show that, in fact, these ionic interactions are indeed broken by protonation, substantiating this model that what protons do is to pre-break some important bonds that have to undergo dissolution during the gating transition. And this provides us and others with interesting mechanistic insights and structural insights that can tell us about how one might develop drugs that are more selective for modulating this channel. Let me end by thanking the many people who have made this day possible. First and foremost are all the members of my lab, past and present, who have contributed to the body of work that I've described and other aspects that I haven't had time to talk about. I've been able to highlight a few of them, but there are many others, their names shown here, who've been such important players in our work over the years. I've also been fortunate to enjoy interactions with a number of fantastic collaborators throughout the world who've helped us explore new areas and models of acute and persistent pain, as well as molecular structure. And in particular, there are two people here at UCSF, Alan Bastbaum and Yifan Cheng, my friends and colleagues, who have taught me so much about the physiology of pain and the biophysics of structural biology, respectively, and it's been a great joy working with them and members of their lab over some 20 or 30 years. I'd also like to thank the National Institutes of Health for being a primary supporter of work in my lab and allowing us to follow our curiosity. And finally, I'd like to thank the community here at UCSF, who has provided a really welcoming, interactive, and friendly home that's encouraged me to explore new areas and has been a fantastic place for my trainees, my fellows, and postdocs to explore their passions, to learn from their colleagues, and to really expand their boundaries into new areas. Now, I'd like to also end by thanking my mentors. As the late Al Gilman once said, who you are as a scientist is a direct reflection of the people with whom you've trained. And I've been so fortunate to work with some really fantastic scientists over the years who taught me much of what I know. I began by working in the laboratory of Alex Rich as an undergraduate at MIT, where I explored mechanisms of tRNA biosynthesis. And here's a picture of me that some friends sent me from the archives at a pop-up party that occurred down the hall soon after I joined Alex's lab for David Baltimore, who at the tender age of 36 or 37 had been awarded the Nobel Prize together with Howard Temin for discovering first transcriptase. And here I am with a lot more hair, standing next to Tom Fraser, who was a graduate student in Alex's lab, who took me under his wing and really first taught me about how to do science in a rigorous way. I was also fortunate to meet Simon Litvak, who was a visiting scientist at the time at MIT, and who allowed me to come to his lab for a few months at the University of Bordeaux to purify enzymes, such as tRNA nucleotide transferase. But I also learned a lot about life, the quality of life, and how to enjoy all things like that, including red wine from the Médoc and other areas of Bordeaux. And following that, at graduate school, I was incredibly fortunate to come under the tutelage of two dynamic and exciting scientists, Jeremy Thorner and Randy Sheckman, with whom I worked to explore mechanisms of peptide hormone processing in Baker's yeast. And from Jeremy and Randy, I really learned the power of combining genetics with biochemistry to go deep into a problem and understand how to attack biological problems in a multi-pronged way with rigor and multi-dimensions. From there, I was fortunate to secure a postdoctoral fellowship in the lab of Richard Axel. Richard taught me many things, one of which was how one can use molecular genetics to explore seemingly complex problems in mammalian physiology. He used to say that everything is clonable, and that if you did so, you would develop new ways of looking at biology and physiology. He also taught me how to give people the freedom to explore, to fail and explore, so that they could do unusual things, and then how you enable them and allow them to take those new discoveries to their own lab to start their own independent careers. And I can think of no one who has a more storied list of trainees and protégés. So many of Richard's people have gone on to fantastic careers of their own. Now, in addition to this, there's also the life mentors, the people who shape our lives every day. And of course, my wife, Holly Ingram, who's also a biologist and a faculty member here at UCSF, has had a major impact on me over the years. We've been married for 25 years. And Holly's a fantastic scientist in her own right. She has an intuitive feel for physiology. And I've learned a lot as a reductionist biochemist from her in how to explore complex physiological systems. But more importantly, we've had a fantastic life together and supported each other in this sometimes challenging but also exciting pursuit of scientific research and mentorship. And together with our son Philip, who's taught us, who keeps us honest and has taught us that science is not the only thing in life and that we need to pay attention to other things. And then there's my parents, who encouraged me to do what I was most excited about and to follow my passions. And my father, I remember, told me that most people go to work because they have to. There are some people on the planet who go to work because they want to and like to, and I should always do something that put me in that latter category, to go to work because I was passionate about what I was doing, and that would be more rewarding than anything else. So I thank all of these people for enabling me to do what I've done here today and to be recognized by this incredible honor. Thank you so much, and I look forward to seeing you hopefully next year in person in Stockholm.
The second lecture is on the discovery of receptors for touch by Dr. Ardem Patapoutian. Ardem Patapoutian is professor in the Doris Neuroscience Center at Scripps Research in La Jolla, California, and Howard Hughes Medical Institute investigator. Professor Patapoutian was born to an Armenian family in Beirut, Lebanon, where he attended elementary and high schools. After graduating from high school, he attended the American University of Beirut for 1 year before immigrating to the United States of America in 1986. Professor Patapoutian received his bachelor's degree in cell and developmental biology from the University of California, Los Angeles in 1990, and he obtained his doctoral degree in biology from California Institute of Technology in 1996, where he worked with Barbara Wold focusing on muscle differentiation. Professor Patapoutian performed his postdoctoral training in the laboratory of Louis Reichardt at the University of California, San Francisco, where he worked on the developmental program driving subtype specificity of somatosensory neurons for touch and pain. Ardem Patapoutian joined the faculty in the Scripps Research in 2000, and he also held a position at the Genomics Institute of the Novartis Research Foundation between 2000 and 2014. Ardem Patapoutian is a member of the National Academy of Sciences and a fellow of the American Association of Advancement of Science. Professor Ardem Patapoutian will now deliver his Nobel lecture entitled, "How Do You Feel? The Molecules That Sense Touch."
It has been such an amazing experience to share the 2021 Nobel Prize in Physiology or Medicine with David Julius. I thank the Nobel Assembly, members of the Nobel Committee for Physiology or Medicine, and the scientists who have supported our nomination. I'm grateful that basic research is being recognized in such a way, as biomedical laboratories around the world not just advance our understanding of life, but are the nurseries for future medicines. Our perception of reality is defined by our senses. We have learned quite a bit over the past decades about how we smell, taste, and see. We know much less about the sense of touch, which gathers mechanical, thermal, and chemical information from peripheral tissues such as the skin and translates into an electrical signal that neurons understand. More generally, most cells in our bodies communicate through chemicals, but cells also respond to pressure, and how this is accomplished has remained unknown. So I'm here to tell you that your sense of touch is amazing. Human tactile discrimination extends into the nanoscale. You can sense indentations 500 times thinner than a human hair. Beyond this, we rely on affective touch for our emotional health. And for the related sense of proprioception, allows us to know where our fingers and arms are in space without looking at them. And this allows you to perform complex tasks such as walking or playing a musical instrument, beautifully demonstrated here by Yo-Yo Ma and Emmanuel Axe. But I don't know of any other image that highlights the power of touch more than this one. So this boy is visiting the White House and tells President Obama that his friends tell him that his hair looks like the president's. Mr. Obama lowers his head and says, "Why don't you touch it?" Of course, we're not just talking about sensing texture of the hair, but the intimate, affective nature of touch. We often say, seeing is believing, but in this case, touching is believing. So for mechanosensation, the flip side of touch is, of course, pain. Acute pain that warns us of danger in our environment is essential for survival. Chronic pain, when the noxious stimulus is no longer there, but pain persists, remains a major unmet medical need. For the neurons responsible for detecting mechanosensory stimuli relevant for touch, proprioception, and pain, mainly reside in dorsal root ganglia, or DRGs, a group of sensory neurons present just dorsal to the spinal cord. So they gather information from the periphery and send information to the spinal cord and the brain. DRG neurons are very heterogeneous. Some are specialized to sense temperature and pain, but low-threshold mechanoreceptors respond to touch and proprioception. These neurons are also characterized by their peripheral targets. For example, proprioceptors innervate muscle spindles, whereas various neural endings that are found in the skin are responsible for the sense of touch. Here I'm simplifying sensory receptors responsible for touch, but different fiber types, such as Merkel cell neurite complexes and Meissner corpuscles, play unique roles for our sense of touch. And the labs of David Ginty, Alan Lumpkin, and many others have contributed to a deeper understanding of this process. In addition, many DRG neurons project to internal organs, such as the bladder, and are involved in a process called interoception that I will talk about later. So how do you sense a gentle breeze or a cactus pricking your finger? We have known for quite a while that somatosensory neurons, for example at your fingertips, express specialized ion channels that are directly activated by physical forces. So over the last 25 years, work led by David Julius, my lab and others have identified trip channels required for thermosensation and pain. However, just 12 years ago, the identity of mechanically activated channels for touch and pain, or really any other biological process that depends on pressure sensing, had remained elusive. For those who are not molecular biology experts, I wanted to provide a scale of how small these sensors are that we're looking for. So a grain of rice is 500 times bigger than a cell. Here's an average cell, about 10 micrometers. An ion channel is about 100,000 times smaller than a cell. They would be sitting on the plasma membrane, but they're too small to be seen with this scale. So what do we know about these elusive mechanically activated ion channels? Hudspeth and colleagues were the first to demonstrate their existence and importance. They showed that signaling was too fast for hearing to be explained by a second messenger system. So they hypothesized the direct activation of a mechanically activated channel to be involved in hearing. Later on, Sachs and group showed that this type of stretch-activated currents are present in many different cell types. So how about the identity of these ion channels? So mechanically activated channels have been discovered in many species, including bacteria, worms, and flies, and shown to play important mechanosensory roles. However, either these sensors did not have homologs in mammals, or a direct role for them in mechanosensation was not forthcoming. Interestingly, mammalian mechanosensitive ion channels that conduct potassium have been identified. However, this could not be the touch sensors, for example, as their activation would inhibit rather than activate neuronal firing. So it was not till 2010 that my lab identified piezo1 and 2, non-selective cation channels from mammalian cells. These are the ion channels I will discuss today. So why has it taken so long to identify these elusive mechanosensors? It would help perhaps to look back how other sensors were identified. So the first transduction molecules identified was for vision. Rhodopsin, a GPCR, converts light into electrical signal. Now 50% of the disc membrane area of a rod outer segment is made out of rhodopsin. And this availability in large amounts enabled biochemical approaches to purify and identify the sensor. It was known that G protein signaling was also involved in olfaction and taste. And indeed, Axel and Buck and Zucker and Riba identified olfactory and taste receptors by homology to rhodopsin family of GPCRs. However, the mechanosensors we're looking for are known to be ion channels, and unlike GPCRs, these channels do not have robust homology among each other. Therefore, we thought a functional genomic screen was the best approach to identify them. So today, I will tell you about how we identified piezo ion channels, and then I will focus on the role of neuronal piezo2 in somatosensation and interoception. Finally, I will touch upon the role of piezo1 in non-neuronal cells. This is Bertrand Coste, a postdoctoral fellow in my lab, who came specifically to identify the molecular identity of these mechanosensors. And now he's a faculty member in Marseille, France. So Bertrand adopted a simple cellular assay to assess mechanically activated channels. So while recording from a cell, he would come with the glass probe and push the cell. And if this cell expresses a mechanically activated channel, he could record such currents. He then took a reductionist approach and to try to find this channel from a simple to work with cell line, so that once we identified the channel, later in vivo work would tell us which physiological settings it's playing an important role in. So he screened several cell lines and found that Neuro2A cells have robust mechanically activated currents. And then he used various bioinformatic tools to come up with about 300 candidates that could be this channel. For example, they would have to be multi-transmembrane domain proteins and would be expressed in Neuro2A cells. And then he would knock them down one at a time using an RNAi screen to find the molecule responsible. So this is the screen that he performed. He would knock down each candidate represented by a single data point, one by one, and he would record maximal mechanically activated currents. To get these kind of error bars for each data point, he spent about 2 to 3 days on each candidate. So after almost a whole year of just getting negative data, he finally found that knocking down candidate number 72 had a profound effect on these mechanically activated currents. So this candidate was called unknown family 38 member A. He quickly renamed it piezo1 from the Greek piezi, meaning pressure. So we got really excited then when Bertrand got these results. So he was able to clone the full-length piezo1, and when he expressed them in naive cells, such as HEC293 cells, he could see robust nanoamp level currents. So this is showing one such recording where the more you push on the cell, you see larger and larger currents. And this is the quantitation for those results. So piezo 1 has a sister. We named it piezo 2, and it's about 40% identical at the amino acid level. And it's also mechanically activated, albeit with a slightly different inactivation kinetics. So one of the unique properties of piezos is that they're very large molecules. Mouse piezo 1 is about 2,500 amino acids and has many transmembrane domains. So all ion channels have to have domains that traverse the membrane, but normally ion channels would have, on average, 2 to 6 such transmembrane domains. Here you can see in orange bars that piezo has 38. That is actually more transmembrane domains than any other protein in your genome. In addition, we now know that piezo comes together as a homotrimer, forming a complex that is almost 1 million Daltons. So Kei Satome in the lab, together with Ward Lab here at Scripps, used cryo-electron microscopy to solve the structure of piezo 1. Similar structures were solved by McKinnon and Zhao labs. So you can see the 3 protomers here shown in different colors. They all come together at the central C-terminus where the pore is. The rest of the protein, you can see each ribbon is a transmembrane domain, extends into the plasma membrane. And they extend into the plasma membrane, presumably acting as membrane tension sensors. You will immediately notice that the whole structure has an unusual upward bend to it. And the current hypothesis, first proposed by Rod McKinnon, is that these channels sit in grooves within the plasma membrane, and that any tension or stretch of the membrane will cause flattening of piezo structure and hence activation. So to complement our in vitro work on the structure and function of these ion channels, we have spent considerable amount of time elucidating their function in vivo. So both in mouse models as well as from human clinical data, we have been exploring the role of piezo ion channels and asking what happens if piezo is not present, loss of function studies, or when we have too much piezo signaling, or gain of function studies. We were very interested when we found that piezo 2 is robustly expressed in the sensory neurons of the dorsal root ganglia. These are the neurons I told you about that are required for touch and pain. This is an RNA scope, RNA in situ hybridization, that shows in brown expression of piezo 2 in a large subset of these sensory neurons. So we have made transgenic mice that do not express piezo and have done many experiments to show that piezo 2 is the principal sensor for touch. I just wanted to show one very early experiment that we did. So these are wild-type animals, and we've put a tape on their back. As you can see, they don't like it and try to remove it. Piezo 2 knockout animals, on the other hand, as you can see, they don't seem to be bothered by this at all. So this work was carried out by Sanjeev Ranade and Sung Woo in the lab, collaboration with Lumpkin and Lewin labs. So I mentioned proprioception before. I think this is perhaps your most important sense, and yet most people don't know that they have it. The sense of where your body is in space is based on mechanosensory neurons that innervate muscle and tendons. Thanks to this sense, you are coordinated, can close your eyes and touch your nose, and with a lot of practice, might be able to do things like this. So is piezo 2 required for proprioception? Work done by Sung Woo in collaboration with Wilkinson and Jessel labs also showed that piezo 2 in the mouse is required for proprioception. Here is an animal that has no piezo 2 mid-thoracic and lower. You can see that this mouse lacks coordination in its hind legs, a sign of lack of proprioception. For all behavioral tests that we do, we confirm the role of piezo 2 in transduction pathway by using ex vivo electrophysiology. In this example, Wilkinson lab can stretch muscles while recording neuronal activity from proprioceptors. As you can see, in wild-type animals, as you stretch the muscle, you see massive increase in neuronal activity. In contrast, in the knockouts, we hardly see any activity. Interestingly, the labs of Chesler and Bonneman at NIH identified patients with loss-of-function mutations in piezo2. And very interestingly, they have the same deficits in touch and proprioception. So in this video, a test for proprioception, one of these individuals is asked to walk in a straight line while blindfolded and harnessed for their own protection. As you can see, she has a very difficult time performing this task, something that you and I can easily do. So how about pain? We know both in mouse and humans that acute pain is relatively normal, but we wanted to ask about tactile allodynia. So this tactile allodynia is a painful sensation that is caused by an innocuous stimulus such as touch. You might experience this after injury or just after sunburn, but this is also a phenotype experienced by patients who suffer from neuropathic pain. So Shweta Murthy in my lab, as well as Alex Chester's lab, demonstrated that piezo2 is essential for tactile allodynia, both in mice and in men, opening a possibility that blocking piezo2 in some instances could be clinically relevant for people suffering from chronic pain. So the requirement of piezo2 in touch, proprioception, and pain entails sensing from peripheral tissues such as skin and muscle. But mechanical force transduction is also relevant for internal organs. So although DRGs do innervate internal organs, there's also a whole other sensory system dedicated to this. So the cranial ganglia of nodos through the vagus nerve innervates internal organs and gets mechanical and chemical information from them, including lung, the heart, stomach, and the gut. Both the mechanism of mechanotransduction, as well as, in some cases, the precise physiological role of mechanotransduction in these tissues is not understood. Over the last few years, work done by Keiko Nonomura, Sung Woo, Weizheng Zhang, and Kara Marshall, and in collaboration with Liberalist Lab and Chester Labs, we have begun to investigate the role of piezos in interoception. For example, lung stretch is sensed by these sensory neurons, and this information is used to control breathing rate. Piezo2 plays an important role in this process. Although you're not conscious of this, there are specialized neurons that sense pressure in your aortic arch, and this information is used to keep blood pressure constant. Piezos are also required for these baroreceptors. On the other hand, you are very aware of bladder stretch as it alerts you when it's time to relieve yourself. Both in humans and in mouse, piezo2 is required for this important sense. And indeed, we found that humans without piezo2 plan to go to the restroom on a schedule to avoid accidents. And I wanted to highlight that the role of mechanosensation in interoception is an active area of research. We are, for example, currently investigating the role of piezos in the gut. When you eat a large meal, you feel very full. That sounds very mechanical in nature. And yet, most of research on satiety is focused on nutrient sensing, for good reason. But we do not know how much stomach stretch contributes to how much you eat, when you eat, diabetes, obesity. We hope that studying these molecules will illuminate the role of mechanosensation in food intake and digestion. So Kara Marshall and Rocio Servin in the lab are currently investigating these related questions. So I've talked mainly about sensory neurons, but many other cell types experience, sense, and respond to mechanical force. For example, blood vessels experience shear force from blood flow, muscles get stretched, and many different cell types get mechanically indented. Not just sensory neurons, but for example, red blood cells that squeeze through capillaries half their diameter. And the mechanism of mechanotransduction in most of these tissues remain unknown. So piezo1, the sister molecule of piezo2, is widely expressed in many tissues, and our lab and many others have only now begun to explore the role in various biological processes and diseases. For example, we and others have shown that piezo-1 is required for blood vessel formation during development. Others have shown an important role of piezo-1 in regulating bone density. Very recently, together with Mehta Pathak's lab, we showed that piezo-1 in the skin is involved in wound healing. Finally, we and others have found various interesting and unexpected roles of piezo-1 in red and white blood cells. I will spend a few slides talking about this. So we have strong genetic evidence that piezo-1 plays an essential role in red blood cell volume regulation. So gain-of-function mutations in piezo-1, both in mice and in humans that have a condition called hereditary xerocytosis, have dehydrated red blood cells, while loss-of-function mutations in mice causes overhydrated red blood cells. So the hypothesis for normal function of piezo-1 in red blood cells is that when they get squeezed through capillaries, the RBCs actually shrink to more easily navigate these tiny vessels. In addition, partly due to the red blood cell dehydration phenotype, Shangma in the lab showed that gain-of-function piezo-1 provides protection against malaria infection in mice. So the next question was, if excessive piezo-1 signaling in the red blood cells is indeed protective, are there piezo-1 gain-of-function mutations in regions that are endemic for malaria? We indeed found a subtle gain-of-function allele, E756 delete, a deletion of a single amino acid, that is very prevalent in African population. So this map shows the frequency of this allele. The wild-type copy is in light blue and the gain-of-function in purple. As you can see, E756 delete is almost non-existent in people of European ancestry, but prevalent in African population as well as in African Americans. And this allele is quite abundant. 1 in 3 people of African descent are heterozygous for this allele. That translates into about 400 million people carrying this allele, with about 40 million people being homozygous. Now, since this allele is a very subtle gain-of-function mutation, it does not by itself cause any disease, but we think it is very interesting to investigate the consequences of carrying this allele. And we are finding out that there might be both advantages and disadvantages of carrying this allele, perhaps similar to what is known about sickle cell disease. So the advantage, as I mentioned, is its potential to protect against malaria. This is mainly shown in mice, and more work needs to be performed to assess its relevance in humans. On the other hand, we have recently shown that too much piezo-1 signaling in macrophages causes increased phagocytosis and iron levels in the blood, which is not good, as iron overload is associated with many pathologies, including organ failure. I will show a single slide on this data. So Shangma received blood samples from more than 300 self-reported healthy African Americans, and this shows the distribution of E756 delete. As expected, about 1 in 3 have the gain-of-function allele, shown in green, and very few are homozygous in orange. However, when we look at clinically relevant markers of blood iron levels, transferrin saturation and ferritin, we notice that within individuals that carry high iron levels, we observe a higher percentage of E756 delete carriers. Indeed, with a more stringent assessment of increased iron levels, all 10 individuals had at least 1 copy of the gain-of-function allele. Now, Shang has shown that these E756 delete carriers are 12 times more likely to have high ferritin levels than non-carriers. Larger studies are, of course, required to assess the risk factor of individuals carrying this allele. And finally, for the future, we are eager to perform more genotype-phenotype association studies to find out other potential physiological processes that depend on mechanotransduction. So till now, I have focused on mammalian piezo-1 and 2. This slide shows, however, that piezos are conserved across evolution. So flies and worms have piezo, many unicellular organisms have piezo, even plants have piezo channel. Plants also experience mechanical force. For example, when roots grow and encounter a harder surface in the soil, they have to decide whether they go right through it or around it. Recently, Ali Reza Musavi in my lab showed that piezo in plants is expressed in root tips. So in Arabidopsis, he showed that without piezo, the ability of these roots to penetrate through harder surface is compromised. I find it absolutely fascinating that the same type of sensor that we use to sense touch is also used by plants to sense substrate stiffness. Evolution is indeed amazing. In conclusion, we have shown that piezo 2 is the principal mechanosensor for touch, proprioception, and many interoceptive functions. We have also shown that piezo 1 plays a predominant role in many non-neuronal cell types. Overall studies in both human and mouse genetics are providing a foothold into unexpected roles of mechanotransduction in physiology and disease, including surprising roles such as regulating blood iron levels. Finally, there are more mechanosensors to be found. For example, for noxious pain, we know that there's a separate channel that accounts for this function. However, its identity is unknown. So the field is ripe for many more exciting discoveries. I have many people to thank for their roles in my scientific journey. I fell in love with basic discovery research in Judy Laniel's lab at UCLA, learned how to ask important questions in Barbara Wald's lab at Caltech, was inspired by Lou Reichard at UCSF to take on high-risk endeavors, and discovered how to harness cutting-edge technologies from Pete Schultz at Scripps Research. Learning from all these mentors, I have tried to build a laboratory environment at Scripps Research that emphasizes impact, collaboration, and collegiality. I hope they are having as much fun as I am while pursuing such science. And I'm exceedingly proud of how well they work with each other and with our expert colleagues around the world. So these are the names of all the scientists who have worked with me in the past 21 years. These include current members as well as alumni. Indeed, one of the greatest joys of doing science is to see the trainees blossom into independent thinkers, chasing important questions both in academia and industry. And of course, we could not have accomplished much without collaborators from laboratories across the United States and beyond. So the importance of funding in biomedical research cannot be overstated. Howard Hughes Medical Institute affords freedom to pursue major innovations and has fueled my research program. The National Institutes of Health has been vital to the very existence of basic biomedical research in the US. During our COVID pandemic, I sincerely hope that the United States, and indeed the world, has a renewed level of appreciation for the impact biology research has on human health. And I want to end with a personal note on this image with my son Luca and me, a few minutes after I received the call from Stockholm and while the official announcement was being made. I'm very grateful that my wife Nancy snapped this photo. I study the sense of touch, and this is indeed a very touching moment of pure joy. My family has been a profound source of happiness and the foundation of my ability to do science. And because this is 2021, with restricted in-person interactions due to the COVID pandemic, this photo perhaps took on added meaning as it spread the news of the prize. And through social media, I received messages from around the world. Indeed, many congratulations came from people of Armenia and Lebanon, where my ancestors and I were born. I am grateful that the Nobel Prize has reconnected me with my roots and highlighted how internationalism, diversity, and the exchange of ideas and people are so integral to progress. Thank you for the opportunity to share my science with you.
I guess you all share my sentiments. This was fascinating, it was touching, and it had a clear address to all of us. On behalf of Karolinska Institute, I would like to thank you both, Dr. Julius, Dr. Patapoutian, for your outstanding lectures here today. Through your discoveries about our ability to sense heat, cold, and touch, you have contributed greatly to our understanding of how we perceive the world around us, and I should add, the world within us. You have identified critical missing links in our understanding of the complex interplay between our senses and the environment. Knowledge that will benefit a wide range of areas in the realms of research and healthcare. So, we very much look forward to welcoming you both to Karolinska Institute and to our beautiful Aula Medica when circumstances allow, hopefully in a not too distant future. And to all of you who have joined us today here in this hall or on distance, thank you for your attention. I'm sure you have also found the lectures very rewarding, and I wish you a pleasant day and an inspiring Nobel week. Thank you very much for your attention.